Today, we're diving into a breakthrough that's basically revealing entire hidden chapters in the story of cancer. For the longest time, we thought we were reading the full book, but it turns out huge sections were completely missing. So let's talk about what we've been missing all this time.
You know, we've always known that cancer is, at its core, a disease written in our DNA. But what if I told you that for decades, we've been trying to read that book with half the pages torn out and the rest smudged? We could only guess at what was written there. So the real question is, what secrets have been hiding in those missing chapters? So, to really get why this is such a big deal, we have to start with this one core idea. Cancer is a disease of the genome. It all starts with changes, mutations, in our DNA. Some of these we inherit from our parents. Those are called germline variants. But most of them are somatic mutations. You can think of those as typos that just build up in our cells as we go through life.
Now, to find those typos, you have to read the genome, right? But for years, our main way of doing that, well, it's been like trying to read a book after it's been put through a paper shredder. And this one problem has created a massive blind spot in our understanding of cancer.
Okay, now don't get me wrong. This method, it's called short-read sequencing. It's been absolutely incredible. It gave us these massive projects like the Cancer Genome Atlas, which cataloged millions of mutations from thousands of tumors. I mean, it literally built the foundation of modern cancer research. But it has a fundamental flaw.
Let's go back to that shredded book analogy. Imagine you're trying to piece a book back together from tiny little strips of paper. Now, if a sentence is unique, like the quick brown fox jumps over the lazy dog, you can probably figure it out. But what if a page just says the same word over and over and over? The lazy, the lazy, the lazy. With those tiny strips, it's impossible to know how long that repetitive section was. And it turns out our genome is full of these repetitive parts.
And because of that shredded book problem, our old tools were basically blind to huge types of genetic changes. We were missing these big chunks of DNA being inserted or deleted. They're called structural variants. We couldn't see complex rearrangements where DNA gets all jumbled up, and we definitely couldn't make sense of those repetitive regions. We even had trouble with something called phasing, which is just knowing if two typos you found were on the copy of the gene you got from your mom, or if you got one from each parent.
So if the old way was like reading tiny, shredded snippets, the solution, you guessed it, is a technology that lets us read entire pages or even chapters of the book all at once.
And it's called, simply enough, long-read sequencing. The name really says it all. Instead of reading tiny fragments of DNA, this tech reads incredibly long pieces. And these long reads can just stretch right over those confusing, repetitive parts, giving scientists a clear, totally unambiguous view of what's going on for the very first time.
What you can do with this is just mind-blowing. From a single blood sample, scientists can get this incredibly rich, multi-layered picture. They're not just seeing the DNA blueprint itself. They can also see which genes are being used and which ones are switched on or off. It's like going from looking at a flat 2D map to exploring a fully detailed 3D model.
Okay, so we've got a new tool that can read the whole book. So what have we found in those chapters that were hidden from us for so long? This is where it gets really exciting. Get this, a staggering 8% of the human genome was just missing from our map. It was a complete blank. A mystery. But in 2022, using long-read sequencing, a group called the Telomere-to-Telomere Consortium finally filled in every single one of those gaps. They gave us the first truly complete sequence of a human genome. And that missing 8% wasn't just a rounding error. No, it amounted to over 100 million letters of genetic code that had been totally invisible to science before. It's like we discovered a whole new continent on our own genetic map, suddenly open for exploration. But here's a really crucial point. This isn't just about finding new things, it's also about being way more accurate. With this new, complete map, long reads slash the number of false alarms in medically important genes. We're talking about reducing errors by a factor of 12 in some cases. That means fewer wild goose chases for researchers, and a lot more certainty for doctors and patients. And that accuracy is already paying off big time. In one study of breast cancer patients, researchers found hundreds of variants in known cancer genes that were just completely invisible to the old short-read method. One of them was a structural variant hiding inside BRCA1, probably the most famous cancer risk gene there is.
This has immediate, real-world consequences. Let me give you a perfect example. There was a variant on chromosome 16 that short reads flagged as pathogenic, meaning disease-causing. Pretty scary. But when scientists looked again with long reads, they saw the whole picture. It was actually a different, likely harmless variation. They were able to reclassify it as likely benign, which could have saved a patient from a world of unnecessary anxiety and maybe even treatments.
So this is obviously transformative stuff. But it brings up the big question, right? How do we get this amazing technology out of the research lab and into the hands of every doctor and every patient who needs it?
Well, there's a pretty clear roadmap. First, the costs have to keep coming down. Second, we need faster, smarter computer programs to make sense of all this new data. Third, we've got to scale up the studies from just a few dozen patients to thousands so we can really see the patterns. And finally, we have to figure out how to weave all this complex information smoothly into the day-to-day work of a hospital or clinic.
The challenges are real, for sure, but the momentum here is just undeniable. We are literally moving from a blurry, incomplete picture of the cancer genome to a high-definition, comprehensive view. So the question we're left with is this. As we finally begin to read the complete story written in our DNA, how is it going to rewrite the future of diagnosing, treating, and maybe, just maybe, one day, preventing cancer itself?